So analogy I like to make are these two birds. So this is a regular protein, polypeptide and a glycan. This is just the polypeptide part. And even if we are the best scientists and have all the money and have a perfect experiment and we do it perfectly, if we are trying to understand flight and we use this bird as a model, this is closest to flight we would come.
Hey guys, really excited today because it's been a few years. Myself and this gentleman, Professor Gordon Lauc, have been very busy. So we kept meaning to get back together on glycosylation of your proteins, aging, longevity, and GlycanAge testing. And here we are, Professor Lauc. Great to see you.
Thank you. Thank you to invite me here again. It's great to talk to you again.
Yeah, and it was great to meet you in Croatia and stay up near you there and have that barbecue, all that delicious meat. We're not anti-meat on this podcast, of course. So, yeah, I don't know. You have a few slides, and just to get me up to speed, you're in a huge conference, I believe. Uh, I think thousands of people in American Medical Association. Maybe give a quick flavor of that. It sounds pretty intense.
Okay, so I can give you a short kind of intro into what I was talking about. And it was a large conference of the American Academy of Anti-Aging Medicine. I had a plenary lecture in front of something like 7,000 people, a bit scary. And
Hmm.
I was talking about glycans. And not many people know what are glycans, so I will give you also a short introduction here to remind people who probably never learned about it. So the majority of proteins, and proteins are the robots which do all the work in our body, are glycosylated, meaning they have polypeptide part that's made from amino acids, and then they have a glycan part. And the key difference between the polypeptide, which is gray here, and a glycan, which is in color, is that the polypeptide is defined by a sequence of nucleotides in our gene. So we got it at the moment of conception, we can't change them. It's fixed. It's changing very slowly through evolution. While glycans are product of networks of dozens or hundreds of genes. So this is a network which we mapped for IgG glycosylation. So it's hundreds of genes, their epigenetic regulation, environment, working together to decide about the glycan structure, which is being added to the protein. So if you think about functional aspects of glycosylation, it's the same as coding mutation. Coding mutation is a change of nucleotide, which is changing an amino acid. It's a change in protein structure. It can kill you. Alternative glycosylation can do the same thing. It's changing the protein structure. But from
And
evolutional or informational aspect, glycan composition is not monogenic. It's a complex trait, meaning it's a number of genes which integrate together, and this enables diversity. This is enabling us to become different from other individuals in our population without the risk to genetic heritage. So we don't have a mutation to make us different. We are becoming different just by reshuffling alleles. And what you see revolving in the middle, it's one typical glycoprotein. In this case, it's the S-glycoprotein of the SARS-CoV-2 virus. And everything here on the surface is a glycan. So if you think that you can understand the function of a protein without seeing anything about glycans, you're probably wrong. And if you talk, for example, about immunoglobulin, the main antibodies in our system, their effector functions are regulated by glycosylation. So this central part is in this constant domain is the glycan, and depending on which glycan we attach to antibodies, they will either be pro-inflammatory, so they will kill the target, and they will be anti-inflammatory. They will ignore the target. And for example, one thing which happened in the pandemics when we made this huge experiment with the billions of people being tested is that with the millions, with the multiple booster doses, all the antibodies were silenced. And now people with the multiple doses have higher chance of actually being infected because our immune system thinks that antigen is a self-antigen. It's not attacking it. So analogy I like to make are these two birds. So this is a regular protein, polypeptide and a glycan. This is just the polypeptide part. And even if we are the best scientists and have all the money and have a perfect experiment and we do it perfectly, if we are trying to understand flight and we use this bird as a model, this is closest to flight we would come, because bird without its feathers cannot fly, the protein without its glycans cannot function. And to understand biology, we have to look in all aspects of nature, and glycosylation is an important part. And one word of caution, people often mix glycation and glycosylation. So glycation is a chemical reaction between glucose and amino groups and proteins. We quantify it as glycated hemoglobin, HbA1c, advanced glycation, end products. It's a random damage. It's taking a shotgun and firing at the protein. Glycosylation is this sophisticated modification. And this is making people different. We showed it on mice. We had 8 founder strains, crossed for 3 generations, and then these second generation offsprings were inbred into strains. And some of these strains became completely different without a single mutation, just a reshuffling of allelic variants. And we also see it in humans. So this is 6 random individuals, different colors, different glycan. It's actually glycans which is making us different. The reason why we are all not the same is because we have different glycosylation of proteins. Again, the same word of caution, glycation is something else. Glycosylation is a sophisticated modification, and glycans are this ultimate layer of molecular complexity. It integrates genetics, epigenetics, environment, into chemical structures, which can be reliably quantified. So we can measure effects of genes, epigenetics, food, microbiome, even what we think, by measuring these molecules. And we analyzed over 200,000 people so far. And one of the first things we learned is that glycans change a lot with age. So these are 5 different cohorts: China, Croatia, England, Scotland, Estonia. Blue are men, red are women. We all change with age. Men age linearly. Women are generally protected in the reproductive period, then have this rapid acceleration around the perimenopause. And we learned that this pace of aging is highly individual, and it correlates with the lifestyle. So if we live an unhealthy lifestyle, and we have these all biomarkers of a healthy life, like high insulin, high pyrogen, HbA1c, so on, our GlycanAge will go faster. Our glycans are aging faster. And then we developed this test called the GlycanAge. GlycanAge is a test which is measuring molecules of glycans attached to immunoglobulins, which are not only biomarkers, they are functional effectors of inflammation. So you're directly quantifying low-grade systemic inflammation, and you can modify it either by lifestyle or pharmacotherapy. And of course, as you know, but everybody else should know, I'm conflicted there. I'm inventor of the test. I'm co-founder of a company. Well, this discount is not valid anymore. It was at a conference, so I didn't change the slide. But I always try just to show people the data and tell them, look at the data yourself, and you make your own conclusions. Don't just trust my word. And for example, there was a large study in Qatar done by the colleagues at Weill Cornell in Qatar. They paid for more or less any omics measurement that can be done on their cohort. Different proteomic, different epigenetic, lipidomic, glycomic measures. And then out of 6,000 traits they measured, they looked which are the strongest correlations with age. And 9 out of 20 were immunoglobulin glycans. So definitely the molecules which contain the most information about aging are immunoglobulin glycans. And also there is a large organization called the Biomarkers of Aging Consortium. They published a perspective article in the Cell, and they clearly concluded that the GlycanAge is the only test which is being commercially used to track responses to lifestyle changes, and that this test associates with multiple diseases. Now the question is, the majority of scientists are using epigenetics to quantify aging. But clinics, if they want to track their patients, GlycanAge is the only test which is being used at the moment. And key difference is that analytical performance of GlycanAge is by far the most superior. Because what we did, we invested over 40 million euros in the last 15 years to develop analytical technology, which is returning the accurate result. And what you can see here, these are different components of my own IgG glycom, which I sent to the lab every few months. So this was not analyzed as a single batch. This is an individual sample sent to the lab. I got the numbers back, then 3 months later, and most of the time there is no change, but sometimes you can see a slight change. And when this change is consistent, this means there is biology behind that. And here it was bad because this is the main pro-inflammatory glycan. It was going up. This one is cardioprotective. It's the one predicting heart attack and stroke. It was going down, meaning the chance of heart attack and stroke was going up. So I knew something was wrong. I had to find out what. It was not easy, but eventually I discovered I had a cryptic root canal infection. There were bacteria growing in my root canal I was not aware of, but this is the known risk factor for all the cardiovascular problems. So once I found it, this thing returned to the baseline. So even the slight change can be monitored because of an analytics which is accurate. If I would be using epigenetics here, then the results would be something like that, just going up and down because of the inadequate analytical precision of the chips, which are being used for epigenetic analysis, because they're not made for individual patients. They're made to analyze large cohorts. And this is an interesting example. This is something we are doing together with Alden Scientific. So they want to have a huge database of people with all the possible omics. And this is a single individual who gave 3 samples on one day, and then 2 days later, additional 6 samples. And epigenetic age went from +4 to -5 in 2 days. And this is not biology. This is just the analytical problem of the technology which is being used to calculate epigenetic age. This is not the problem if you are doing a cohort study. You have 1,000 people, and then you have a random error. This will level out, and you will get a biological signal. But if you monitor yourself, and then this was your age at the beginning, and then you did something, and then you get this age, you'll make a completely different conclusion if you get this number.
Yeah.
And this is just random, this analytical variation. So this is the key problem with epigenetics today. Some labs, some clinics are trying to measure 3 or 4 times the same sample, but then this is becoming very expensive. So it's generally clinics don't use this type of approach. And the second key question is, when you measure something, what does it mean? So what is the meaning of the biomarker you measure? And we are trying to understand what is GlycanAge actually meaning. And what you can see here, for example, this is again me a few years later, and there is this rapid acceleration step. This is when I had COVID. I had pretty severe COVID, and my GlycanAge increased for 4 years and never came back. So I kind of lost 4 years of GlycanAge because of this virus. And in large studies we did with the Karolinska Institute, we showed that these glycans associated with higher GlycanAge are actually predicting both mortality and dementia, which is not good. Haven't published this yet, but we'll publish it soon. But what we have published, we looked in 27 populations all around the world, and we saw that people who live in countries with a longer expected lifespan, like Italy or England, have their glycans moving much slowlier toward old age than people living in Uganda or Papua New Guinea or China, Turkey, Thailand, countries which have a shorter expected lifespan. This is population level, this is not individual, but it's a strong indication that faster aging glycans are predictors of mortality. Higher GlycanAge was a predictor of severe COVID on top of age. And interestingly, if you had a severe COVID, if your GlycanAge further increased, it usually had a fatal outcome. So these people with the rapid acceleration of GlycanAge, they just disappeared. They died when this happened. So there is a lot of information in glycans. We analyzed dozens of studies. There is a beautiful reviews if somebody wants to read with all the changes in different diseases. It's a lot of information there. And also, many of these changes resemble accelerated aging. This is the effect of aging, the last 4 bars. These are different diseases. So healthy people look younger when you're looking at their glycans compared to people with many different diseases. And what we actually believe here is that we are quantifying low-grade systemic inflammation, which is contributing to these diseases. And we can see changes in glycans up to 10 years before the disease is first diagnosed. So you're looking at the molecular changes, and then actually you have 10 years to kind of prevent disease if you try to change it. And this was my introduction. And I know it's a tough topic, but I believe it's interesting. We'll invest a lot of effort into research, and I hope we are learning as much as we can.
No, that's a great summary and super speedy too, so we can get into a chat about it. One thing I noticed there was the women in Estonia, it looked like the classic women increasing in aging and diseases, after the menopause. And some people say, oh, it's the hormones, and then they tried to overcome the hormonal effect and nothing happened or it got worse. I heard another one I'm interested in is the loss of iron from the cycle. When that stops, the women increasingly get high iron like men, and that may have an interaction. But Estonia, it looked like, unless it was a small sample, it looked like the women had less of a knee in the curve. The women were more steadily aging like the men.
I think it was a smaller cohort, so it
Yeah,
wasn't so
might
clear.
might
We
mightn't
always
have
have
showed.
very clear acceleration in perimenopause. So the estrogen is a very strong effector. We have really good placebo controlled randomized trials showing that estrogen is working. We never really properly addressed iron. Because what you are saying is true. Yes, they're not losing iron anymore, so it could be iron overload. Maybe we'll have a look. We'll have a look.
Oh yeah, it's interesting. There's a book by a friend of mine, Dumping Iron instead of Pumping Iron. And he goes through in a book all of the evidence. Now, it's not RCT, a lot of associational evidence, a lot of mechanistic evidence supporting excessive iron being a problem. But like you would be, I'm very conscious without an RCT, you don't have any proof, but it's intriguing. So, uh, the other thing, I just want to pick up on a couple more things. The epigenetic is very disappointing because signal to noise there. I mean, epigenetic at a glance, I say, what does it tell you when you're in stage 4 melanoma cancer? Or like, how could you be?
So I wouldn't say that epigenetic is disappointing. It's just analytical methods currently being
Okay.
used to quantify. So there is a room for improvement. So the one of the problem with all this testing of biological age is that most of companies just develop tests to be the most profitable it could be. So they try to make a quick money somewhere. And we are trying to go slowlier, and we are not doing it just to make money. We are actually doing it to develop new technologies to really help people at the end. And it's, I think in the long term, this is good. In short term, we are not really profitable. We are not making a lot of money, but I don't think that the money should be the key driver.
Absolutely. And no, it's great that it is economically solid enough to be able to reinvest, and that should pay off in the long term. So, yeah, I like the approach, but the cut and run is more for like Merck and Pfizer and those guys. So, I actually got my results, and this was when we met in Croatia. You did a kit with me and took the blood. And it was remarkably easy. It was just like your normal kind of finger prick stuff. Nothing, you didn't need large amounts like some testing that does advanced test, like pipette type things. But I got the results and I was delighted as we chatted briefly at the time. Then we both got too busy. So I have them here and I might share them actually, which is very kind of open of me and revealing to everyone. But then again, you would say I'm okay to do so because they're good. Now, I was surprised, Gordon. Um, I was surprised because I think one of my unhealthy lifestyles, if any, though at this time I had been on a few months on a relatively good food diet, I was trying to fix my indulgence in the COVID craziness. And it was actually in a good period when I was in Croatia, but I was still probably drinking too much alcohol, wine and even beer. So I was surprised that the results were quite good. But as you said, carbohydrates, sugars, and all a load of things you'd expect to age you, they really affect the glycan age. And if you change your habits and get another test, you can show improvements. So it's fantastic. But alcohol doesn't seem to show up.
Yeah. So we have zero evidence that alcohol is doing any damage. So I'm not saying alcohol is not doing any damage, just we're not seeing damage on the immune system we are kind of quantifying. And I'm not too much surprised there because, we evolved with ethanol. Eating ripe fruit, there's ethanol in there. And, evolutionary, we were eating fruit when it fell to the ground. So it was already fermented a bit. So we all have this alcohol dehydrogenase. We can remove alcohol, ethanol. Ethanol is not the problem. I think big part of alcohol issue is the methanol, meaning drinking a bad alcohol, and then you have methanol, which is being then oxidized to formaldehyde, which is the bad molecule, because ethanol goes to acetaldehyde and then it's being metabolized. So we are not seeing any effects of ethanol in our large studies. What we are seeing is that people are different. So not everybody responds equally well to different food. And I think this is one of the reasons why we have so many different theories about what is healthy. From some people claiming you have to eat high fiber, plant-based diet, and some are really convinced that this is healthy. And I guess it may be for somebody. But I think for the majority of people, carbohydrates are toxic because in evolution, we never had a large amount of carbohydrates, never until 50, 60 years ago. So there can be very small proportion of people who can really cope with a high carb. And I think the huge mistake which was made 50, 60 years ago, when the food pyramid was made in a way that you should eat a lot of carbohydrates and kind of reduce meat. And if I remember correctly, you're the meat guy. So I hope
Hey.
you're not overloading yourself with the carbohydrates. So I think this is one of the reasons why you got better.
Hey guys, just a brief break from Professor Lauc to let you know himself and his company have expanded hugely since I spoke to him, I think back in 2021. And they've featured widely in mainstream press and also in all the major magazines. And their test is now available pretty much all over the world. So they've offered me a discount of 15% for followers, should you wish to avail. And it is on the more expensive end because of the nature of the technology. But as you saw from my results, it gives a lot of insights and motivation to change what you're doing and then go back and get yourself younger. So in any case, the discount is there should you wish to avail. And let's get back to Professor Lauc. Yeah, I would guess as much. And for around 12 years, I was very good. Obviously, I was lecturing on insulin resistance, hyperinsulinemia, blah, blah, blah. And I went deep into cholesterol and all the metabolic science. So that I was being good. In the viral period for the first year, I was in quite a pressured role because I was going against the machine, or I think as you coined it, the pandemic industry. I think that was your phrase. And
In
but
pandemic industry or pandemic mafia, these two words I use.
yeah. And now all the data's out, we look back, and it's great that many more people are aware that that whole thing was kind of stinky. But in any case, we don't want to talk about that because YouTube kind of demonetizes and you're not allowed to talk about that. Yeah. But those few months before I went to Croatia, I'd been very good and I was getting back on track. Otherwise, I might have got 41 or 43 years, or who knows, because I didn't get a second test, but I will be getting one.
You
And
should. Because
yeah,
the key value of the GlycanAge test is that it's giving you a relatively quick feedback. So it's not rapid. So it takes months to change. So if you do something bad to yourself yesterday or 2 weeks ago, it will not change your GlycanAge. But it's a kind of a steady, quantifiable measure of low-grade systemic inflammation. And maybe what you're saying, you were not that good in pandemics, but if you're really good 2 years before, maybe then you would be 25. And then you got worse to 30 something. Maybe now you're lower. I don't know. Because,
yeah, no, I'm going to have to do it because, and the only reason I didn't, because I had every intention, as you showed, of continuing my good behavior, actually increasing the good behavior, incentivized by the result. Okay, usually you might have more psychological incentive if you get a high age. You might think psychologically, but even a low age, there's an element, like you said, of how low can you go, and also are you staying on track, or like coronary calcification, you might not be able to lower the coronary calcification score, but different mechanism, it's scar, but you can verify that it's not increasing and that you're in good shape, you're not progressing the disease. But yeah, I'll have to get another. I'll just share it for a moment again, just because there's other readings in it I never really got to discuss with you. But just very briefly, so the overall age, of course, and then there's, oh yeah, youth, median, and shield. Maybe very briefly these.
so first we have to be very clear. So the GlycanAge itself, it's a modeled number. This is something what we did as an ultimate simplification to make the number understandable to people. If you tell them you're 5 years younger, you're 5 years younger. But this is the model number. There is no gold standard. Nobody can say this is the accurate number. These indexes, which we named youth, median, shield, they're actually quantifying the structures you see next to the name. So glycans without galactose, with a single galactose, with two galactoses. So these are the molecules which decide to which receptor will IgG bind. And this is hard science. So this is quantified molecular structure with a defined function. And then something we call the glycan youth. So it's a glycan which is kind of quantifying more or less changes in age. And then what you can see on your slide that you are in the green, meaning you are better than the average person of your age. Then you have the glycan median, which is, well, in women, it's the best predictor of heart attack and stroke. In men, we are not fully confident because sometimes we see it, sometimes we don't. So I will not tell you that you are bad, but you are a little bit below median. And this is something which is not really changing with age, it's changing with a lifestyle. So this could be the toll of pandemics on you. It's usually affected by the psychological state of a person also. So if you're under a lot of stress, usually busy executives get bad here. And also the people fighting the pandemic mafia probably get bad there. I'm horrible. So, and then you have this below one, it's a salicylic acid. This is the molecule which is making immunoglobulins anti-inflammatory. So it's a suppressed inflammation. And here you're good again. So it's but something we have to be very clear about is that there is also significant genetic component. So something like 40% of your position is determined by your genes. And this is something which is making it harder to change. But it still means that you are having either higher or lower risk just because of a genetic reason. And actually, if you are bad from a genetic reason, you have to try much harder to actually fix it with a lifestyle than if you are lucky and you have a good genes and then your glycans are good, and then you can be even a little bit less cautious with your lifestyle.
Yeah, I was going to actually say that. Yeah, if the genes are not positive, it's just more incentive. And I think a lot of people where, well, especially on the west coast of the USA, the fitness nuts, if they get good genetics, they probably become obsessed with being extreme in the lifestyle to try and live forever. So you do get that effect in people too. I'm working on a whole psychology program at the moment. So sorry, that's why I keep thinking about psychological aspects of this.
But often we see people who are trying too hard,
Yeah,
doing too much, and like with drugs, there are interactions we don't fully understand. So you cannot, for example, go for ozone therapy, be on a keto diet, and go on intermittent fasting, and take some supplements, and then some people it's just simply too much for them. So
yeah,
I think we have to appreciate more that we are different, and to whichever specialist we go, even the best clinics, they don't really have good tools to stratify us. Things like nutrigenomics don't really work. You can't learn much from your genes. I did my whole genome. I didn't really learn much from it. So I think we have to play with ourselves. So we have to do our own experiments, check what is working, and instead of waiting for 10, 20 years to see the outcome, we can do some of the tests which help, and the GlycanAge is helping. So it is telling you in information which you cannot get anywhere else. But then again, I am selling the test. Don't take my word. Read our
hey,
papers.
I'm genuinely honest. No, I'm genuinely independent on this. And I remember once I worked with an outfit and I kind of discovered that their equipment and testing wasn't actually really that reliable, and it was based on signal to noise actually and some other factors. So you got when you understand, you got to call it. But this stuff it's kind of dripping with genuine bona fide science. It's not the kind of take out your stool sample, do a microbiome, and some charlatan will tell you have to eat grapes all day. I mean, it's the opposite end of the spectrum. You were over there in front of thousands of people in the aging society. So, in fairness, I'm not blowing it up, but this is the real. And I'm not just biased because I got a good result either. I'm not just boosting myself. You know what? We'll really super share the other things. We might as well finish them off. But at a high level, if people are interested in finding out all of this, well, there's plenty online, I'd say, and on your website. Okay, here it is. Think it's sharing again, yeah?
Yeah, now I see it again.
Yeah. Oh, yeah. So just really quickly, high level, people can check it out separately. But
Two additional indexes, which is called Glycan Mature. We kind of made a gentle name out here. It's old. When you're getting old, these are the glycans you get. People get older, they have more of these glycans. You're still pretty good. And the glycan lifestyle is the one which is most responsive to your diet and physical activity. We don't really know what is the function of that specific glycan. We still have to do more research to understand it, but we know it's a kind of indicator of lifestyle. This is why we called it lifestyle. So people really trying hard, they improve this one first.
gotcha. That's the kind of the bellwether or the signal to most watch.
Or for
Okay,
example,
I'll
this
just
one is
see.
highly sensitive to smoking. We can see if somebody is smoking or not.
Yeah, and I guess, if someone really suddenly increased their sugar content or refined grains, I mean, some of the classics that show up probably pretty quickly, I would guess.
Yeah, it would. It would.
Yeah. And here we have, oh, this is informational. Again, I'd say
No,
this
it's
is,
just informational. This is not your
yeah.
personal.
Okay, I'll shut that down then. If you're comfortable, and you are discussing it, you had a relatively surprisingly high score. Now, I love the fact, again, I'm thinking about psychology and stoicism and, being realistic in life. It doesn't bother you too much, but also I think you indicated to me that you kind of know you could improve your lifestyle, but everyone's individual on that too. You're not particularly pressed to do it. But what rough, yeah.
So my glycan age is 80. I'm
Hmm.
54, so it's horrible. But I more or less know what is my main problem. I simply lived too quickly. I had three kids before I was 25. I became professor at the age of 26. So it's had two jobs all the time. And we see it often in busy executives. So I kind of accepted that I will live maybe 5 to 10 years shorter. If I would live a different way, then that would not be me. I cannot live
Yeah.
slow. So maybe,
Oh,
but the good thing about GlycanAge is that whenever you decide to change, you can improve. We've seen people who were really kind of horrible improving for decades after they retire. So
hmm.
it's not that you are kind of doomed. Maybe at one point when I decide, I'm not trying to change the world and introduce this glycan analytics on a global level, maybe I will go and in my orchard grow apples, oranges, and tomatoes, and then I get better.
Well,
And I probably eat less. Yeah, I think
yeah.
carbs are my problem. I eat too many cakes.
Yeah, it's tricky, and yet again, there's I was fascinated as I've done hundreds of hours on cognitive therapy and many other things for a new project, and I was just fascinated because I have so long in weight loss and metabolic health, how much psychology there is in it. In your case, it's kind of very solid and understood by you as you described, but in most people's cases, they don't actually understand the makeup of their brain, their psychology, what drives them to eat. And if they only understood that we have two brains and they have very different kind of goals and drives, it would really help them. So that's my new thing. Anyway, I'm not here to talk about myself.
But that's not
So
that's not easy what you are saying, because for billions of years, only those who ate survived.
yeah,
And now you want somebody to block this primordial drive to eat. And you see how strong it is because a stupid molecule like semaglutide, which is blocking the signal, is actually working.
yeah,
People are losing weight the moment when you're not changing any metabolism, you're just changing the information signaling in your body, and it works. But then you get off the drug, and then it's all the same. So,
boom.
yeah.
But I'll just briefly say it, just because you said that, absolutely true. The fascinating thing is there is the physiological, as you mentioned, the glutide, but the real thing that just stunned me, it's something I sensed and developed in myself a way to deal with over 25 years of pretty stressful corporate, but I could be lucky genetically, hence I don't have the corporate guys high level. Um, but I used to call it my monster. So I knew there was something in me that raised negative thoughts, that ran things down, that procrastinated. And I just sensed it was something separate to me, and I called it my monster. And I used to joke with people when I was flying high and I was really doing well, everything was good. I used to say, I got my monster in the basement. And sometimes I go down and I poke it with a stick if I'm feeling really cocky. But I know it's dangerous because it can make you miserable. And I read books recently, and we have two thinking brains, and one of them is the limbic and the orbitofrontal complex, and that's basically our chimp brain, very similar to the chimp, and we have our human brain, the prefrontal, and they literally are two different thinking machines. And our human brain thinks with facts, figures, capable of compassion and logic and problem solving, etc. And the chimp brain, unfortunately, can only think with emotions, and it has no sense of time. So bad emotions in the past, that's one of the problems. They can come up again because the chimp brain has no time access. But the human brain, funny enough, the human brain can successfully overcome the urges because it's not coming from our human brain. The problem is you need to understand the chimp brain and how to manage the chimp brain. But that's a long story. I won't quit.
You call it chimp. It's not only chimp. It's everything going back probably to basic animals, smallest, simplest animals. So it's just survival. And again, people are different. Some people can use their prefrontal cortex to control the limbic system, but some people can't. So it's very hard to generalize.
Absolutely. It's just there is a very large potential to improve the control, and some people do amazingly well, and some people find it very difficult. You're absolutely right. But oh yeah, it's primordial. But it was Professor Steve Peters, who was he's a consultant psychiatrist, and around 25 years ago, he created that as a model. It's not entirely,
Fine, it's good.
yeah, just so people could visualize to help them kind of be able to manage it as best as possible. It's a great visualization, and it's essentially kind of true physiologically, but you're right, the whole brain is so interconnected, it ain't as simple as that. Yeah, so super stuff. We went off the track there on the GlycanAge. So any other key points you think, or actually has that largely covered it, or any other thoughts?
So I think the other important thing is maybe the future. And I think where glycan profiling is definitely going into routine diagnostics. So I hope in less than a decade, it will be a common test in any biochemical laboratory. So now it's still a little bit exotic test, which you can do in some kind of few labs in the world. Until recently, we were the largest lab. Now we started two labs in US. We are starting some other labs in other countries. So there are more labs, but I hope soon there will be 1,000 labs. And then it will just be one of the other routine tests to do. And then it will not be so exotic anymore. But for now, it's still a little bit exotic.
It is a little exotic, and I often say when I share, these kinds of things to people that are very valuable. In fairness, people of means can try out the more exotic, more kind of very interesting modern technologies, but then many people, in fairness, will have to wait till it scales, and there's economy of scale on the cost, yada yada yada, and that's absolutely fair.
This is not so expensive test.
Not
People
not
people
really,
spend
just
people spend much more money on much less beneficial things. People spend hundreds
yeah.
of dollars per month on supplements, and most of them don't actually help them.
Absolutely. And at the risk of being slightly controversial, um, I noted once I was pressed by many people many times to just consult, not medically, I'm not a doctor, but to consult on the science of their situation, whatever. And I've very rarely done it because I'm too busy. But you just look at the price, cardiac consultant charges, and you realize, like I was going to say, with all due respect, but in many cases, they really don't understand chronic disease, the food interactions, insulin resistance. They have a very narrow view and a heavily drug and surgery kind of centric view. So then when I think of that, I just think, yeah, the amount of money people pay going to specialist this, and then they bounce them to specialist that. And all the specialists are never putting together or integrating the engineering integration of the picture of that person, including all of the data, and giving a proper path forward. Frustrating.
Yeah, it's horrible, but it will get better eventually.
A perfect positive note to let you get back to your day. And that's super to hear that you're expanding and that this thing is really taking off, because you wouldn't like to earn that high GlycanAge result without getting the whole thing to
Without
go, right?
achieving something. I agree. Oh, it was
Great.
great chatting with you again. Thank you.
Absolutely. And we'll circle back later because, as we say, I get another test and find out how bold I've been.
You have to do another one. Yeah, good.
Will do. Thank you so
Nice
much,
talking to
Gordon.
you.
Bye
Bye.
now.
That'll do it, folks. As mentioned, there's a discount of 15% if you want to invest in this technology. I myself was not sure what age I'd get. I was suspecting, because I've been pretty good for a couple of months before it, that I might come in pretty low, but maybe just by a few years. So I was delighted with that, and I really need to go back and check it again. So this test now is pretty much available worldwide. It's been featured widely in the press and in the various health magazines. So the guys really have established themselves and honed it. So if you're interested, discounts there. Again, it's on the higher price end compared to basic simple blood tests, but then it gives you a whole lot more. So that'll do it, folks.